So good morning, everyone. If we can, wow, that actually went easier than I thought. So, good morning and welcome to Brain Day. It's my pleasure to be allowed by Chris to make the introductions for our speakers today. Brain Day is a tradition now. I guess it's 8th year or something like that. 8th year going on, and we have once again a really stellar group of speakers who've been nice enough to come here and join us for the day. Brain Day is sponsored by, well, I guess indirectly by the Faculty of Environment, since they're letting us use this room, but mostly it's sponsored by the Faculty of Arts, the Center for Theoretical Neuroscience, and the Cognitive Science program, who provide most of the underwriting for the activities for today. There should be, cross my fingers, there should be sort of coffee during our breaks, but for those of you in the audience, there isn't a lunch for you. If you're not from our campus, there is a little sort of cafe right next door. You can basically go out this door, and there's another little sort of cafe in the Modern Languages building. And if you want to go farther afield, there's lots of sort of cafes and restaurants if you get all the way to University Plaza. There's the washrooms are right outside the door. Our basic plan will be for about an hour or so for each of the speakers. Then we'll have a short break of sort of 20-30 minutes in between them. And then there'll be a reception following the last speaker up in the Center for Theoretical Neuroscience, which you're all encouraged to join us at and have a chance to maybe follow up with some questions or a chance to talk to some of the people who presented today. I think that's all the sort of procedural news that I have. I'm looking to see if I'm being signaled I've forgotten anything, but I don't think I have. So our first speaker today is Dr. Elizabeth Phelps, and she is sort of extraordinary and exemplary in a number of ways, but one way I think is she's the only one who wasn't an undergraduate at Harvard, but of our four speakers today. But she got to the Ivy pretty quickly after going to Ohio Wesleyan. She went to Princeton, where she did her graduate work. And then she had another Ivy experience because she went to Yale as a faculty member at the beginning of her career. And then she went to NYU, where she has spent her career since then, at least academically, and has worked her way up to where she is now the Silver Professor at New York University. And when you get a chance to do these things and you sort of start sort of scrutinizing people's little online histories and things like that, you get a chance to see and learn some things about people a little more fully. And one of the things that I enjoyed in sort of reading about Dr. Phelps was that, so she's obviously a stellar scientist, but that's going to become clear because she's going to tell us all about the science that she does. But one of the things that I liked is it looked to me like she captured so many of the other elements of what is sort of really, I think, an exceptional academic. So she does service for our discipline. She's president of the American Psychological Society. She's reviewed and been on review boards for numbers of journals. She's part of the editorship of the Cognitive Neuroscience series that comes out, Attention and Performance. I think you're doing that one this year, or just did that one a couple years ago. Okay. I also like the fact that she teaches introductory psychology, so you would think that as you sort of moved up and became sort of a little more established in the department, that one of those classes that maybe was a hard one to teach and a big one to teach is one that you might use your opportunity to sort of get away from. But both in our department and apparently elsewhere, people who are senior accept that it's not just a lot of work, but also a great opportunity to sort of begin the education of new psychologists and not only give them knowledge, but sort of Dr. Phelps is emphasizing that it's important for us to instruct them in how to be critical thinkers, how to evaluate psychological evidence, psychological data. So even if they don't become psychologists, they become better consumers and participants in sort of this process. And then as you look at who she collaborates with, you see that even though there are a lot of very successful scientists at NYU, she collaborates with a great many of them. So she's not just sort of siloed in her own little group. So I know the name Dr. Carrasco, who's a very large name in attention, and Dr. Phelps has collaborated with her, and Dr. Ledoux in emotion, and I think we may hear some more about that. Collaborations there, Dr. Daw, decision making. So in all these ways, she sort of reaches out of her own little confine to sort of participate and collaborate. So the things of sort of service to the discipline, sort of broad participation, collaboration, responsibility for education, all of those things I hope we don't omit our recognition of as we're sort of admiring the science that she's going to share with us shortly. So, Dr. Phelps.
So I want to thank you for having me. It's an honor to be here. And I have to say, when you started to say, in addition to her science, I was a little worried where you were going to go, but that was good. So I want to talk to you today about my work on emotion. And before I start telling you about the research, I want to tell you a little bit about my approach and how I came to this. So I became interested in emotion essentially when I started my postdoc years. I really thought, I want to move. I did memory research as a graduate student. And I thought that when you think about memory in everyday life, the memories we have that persist are things that matter to us. They're the emotional things in our lives. And so this got me interested in emotion. And I also I also decided I needed to learn more about the brain. If I was going to study the human mind, I really needed to understand the brain as well. And so at the time, I wanted to learn more about neuroscience and emotion. There wasn't a lot in humans, but there was some good models in animals, non-human animals. So I started to look into this research, and this is sort of a trajectory I've taken throughout my career, is whenever possible to link back to animal models, because in spite of the fact that we have a lot better techniques than we did 25 years ago to study the human brain with functional imaging, there's a lot you can't do in humans to understand the specifics of neuroscience. So I was inspired by animal models, and this is sort of when I started collaborating with Joe Ledoux, who I'll talk about today. And my work has taken this trajectory where I look to see what I know about the brain system in animal models. I try to, as much as possible, see if we can find evidence for similar neural systems in humans, and by humans I mean undergraduates, using tasks that are as similar as possible. But then ultimately we don't really care how much the human brain looks like the rodent brain, right? We really care how does the brain in humans work in the situations we care about. So then I try to take these systems that I can see, sort of work similarly across species and say, how do they play out in the complex social and emotional situations that are typical of human experience? So I've taken this trajectory throughout my career, mostly making the link from animal models to humans through collaborations, and also here through collaborations with social psychologists and economists. So today, the work I'm going to talk to you about today is more focused on this link here, sort of going from the animal model to the human, and a little less of the social psychology and decision making today. And when I started to look at models for emotion in the brain, probably the most robust model out there was a model of fear in the brain. And so I really started with fear. It wasn't an inherent interest in fear per se, but there's a number of good reasons to start with fear if you're interested in emotion. One is that fear is in some ways one of our most important emotions, in the sense that we learn that things are critical for our survival, the threats to us in the environment. Fear is the basis for many emotional disorders, anxiety disorders, PTSD. So perhaps if we can understand fear in the brain, we can start to think about ways to address those disorders. And practically speaking, fear is one of the easier, at least a little bit. When I say the word fear, you think terror. Obviously, I'm not doing that in the laboratory. But I can make you a little nervous in the lab. And that's a little bit easier than making you a little happy. So it's one of the easier emotions to evoke in the laboratory. And importantly, there's similar mechanisms for expressions across species. So if I want to make the link from the animal model to the human, I can look at things like the fight or flight response, the autonomic nervous system response, which I know operates similarly across species. So all these things suggest this was a good way to start thinking about emotion in the brain. And when we looked at the animal models, there was quite a bit of research based on simple associative fear learning, much as Pavlov described, classical conditioning. So here, this is a slide I borrowed from Joe Ledoux, who I've been collaborating with for the last few decades. It's longer than I want to admit. Here you have a rat that hears a tone. It just sort of looks at the tone, doesn't think much of it. You then pair the tone with a mild foot shock. The tone itself then comes to elicit a range of responses consistent with fear. such as autonomic nervous system responses, freezing, and stress hormone release. We know from lots of work by Joe, Mike Davis, Bruce Kapp, Mike Fanzelo, a bunch of really fantastic scientists, that the amygdala, here in red, and this is going to be sort of the star of our story today, and the pictures I will show you look something like this. The amygdala is necessary for the acquisition, storage, and expression of this simple type of associative fear learning. And the amygdala gets its name from the word for almond, as you can kind of see here. So our very first studies were to bring in healthy humans or undergraduates into the laboratory, show them something neutral. We used visual stimuli, a blue square, pair it with a mild shock to the wrist. After a few pairings, we can start to see responses consistent with fear. So the main one I'm going to talk about today is autonomic nervous system arousal as measured by the skin conductance response. This is a slight change in the electrical conductivity of the skin due to changes in the sweat glands. And what we found, not surprisingly, using functional magnetic resonance imaging, is activation of the amygdala. When I showed you now a blue square that had been paired with shock versus a yellow square that was never paired with shock. Here on the top, this is what a sort of one trial would look like for a healthy control subject. So here is the skin conductance response to the shock itself, the unconditioned response. Here's the response to the blue square paired with shock, the conditioned response. What we saw in patients with amygdala damage was something more like this. So you see this normal unconditioned response, but you fail to see evidence for this anticipation of getting a shock when you see the blue square. So this told us, perhaps not surprisingly, that like other animals, this physiological expression of this learned aversive response in fear conditioning was similar in humans, had a similar neural basis. So that was really the basis of our work. But when we started to look into this, this is a patient with bilateral damage to the amygdala. And here, this is sort of a cartoon version of her data. We showed her her real data, and we asked her to comment. So I want to play for you what she said.
I've noticed if there's anything odd about my reactions to normal lifestyle, life threats, life fear element seems to be, to me, in my head, normal. I've never noticed anything odd.
And how do you feel seeing these results?
I'm dead. I think that it's probably absolutely correct. I know I don't sweat. I knew that there was an anticipation that the blue square was going to, at some particular point in time, bring on one of the bold shocks. But even though I knew that, and I knew that from the very beginning, except for the very first one where I was surprised, I really don't think that I mean, I knew it was going to happen, but that was my response. I knew it was going to happen. I accepted that it was going to happen. So I learned from the very beginning, it's going to happen. Blue shock happened. I turned out to be right. It happened.
So I just want to point, this is Kevin Labar in the background. He was a graduate student in my lab. He's now a professor at Duke who worked on these studies. So as she kind of dramatically demonstrates, in spite of the fact that her body's not telling me she's anticipating this shock, she has perfect knowledge for what happened. We know from other work in our labs and other labs that if I damaged the hippocampus but the amygdala was intact, they wouldn't be able to tell me that it's about this association after the fact, but they would still show this physiological response. So this is just showing me that there's two different memory systems. One's necessary for this conscious recollection of the events that occurred, another that underlies the physiological response that you get when you pair something with an aversive event. But this made us start to wonder, I mean, she doesn't do odd things. She doesn't do dangerous things. So it made us start to wonder, how much does this simple system that we can study in non-humans have to do with the typical fearful situations in humans. So let me ask you, does anybody know what this is? No? See, I don't know either, actually. I got this from a website to teach children about germs. And germs are an interesting type of fear if you think about it, right? Germs are, our perceptual systems don't pick up on germs, right? But you know about germs. People, you can see some people, especially in Asian countries, wear masks because they're afraid of germs during flu season. There was a flu epidemic at NYU, in New York, in the States a few years back, so NYU put this in my mailbox to remind me to keep my hands clean. And germs are really, when we think about our fear of germs, it's completely socially communicated, right? You learn about germs from being told, to avoid germs, that germs can hurt you. And this is just a great example of the social communication of fear. And this is actually quite adaptive. As human beings, we do not need to experience aversive events to learn the threats in the environment. I can tell you what to be afraid of. My puppy can't learn that by me telling her. I can show you, through examples, you learn not to do things because you see bad consequences for other people. So just to give you an example from my own life, this is my mother. I love my mother. I'm not afraid of her, just so you know. My mother's a marine biologist, so here she's sitting with a bunch of seaweed in front of her. So when I was about 10, we went to the marine biological laboratories in Woods Hole. And Woods Hole is right, it's at Cape Cod, it's right next to Martha's Vineyard. There was a movie that came out that summer that was filmed in Martha's Vineyard. I don't know if you've seen it. This one, right? And my mother, being a marine biologist, loves all sea creatures, right, including sharks. She's not really afraid of things in the water. And so, she thought, well, we're here. It's a movie about fish. We should go see it. So, I was 10, went to see this movie. And ever since then, I've had this little tiny fear of sharks. It's not a big fear. I don't stay out of the water. It didn't happen. I have a big brother. It didn't help that we'd be in the ocean and be swimming afterwards, and he'd come up and grab my legs. Didn't help at all. But anyways. But this is a very interesting fear, right? I never had a bad experience with sharks, ever. Yet, even to this day, if I go in the water, sometimes I swim and it's fine. Other times I'm thinking about sharks. I'm like, maybe I'll get out. So it's a classic example of the social communication of fear. And I think as human beings, this is how we get most of our fears. Most of the time, we aren't physically hurt. Most of the time, we're afraid of things. Yes?
Now, I was just wondering if the patient or the subject, you was the subject.
Yes.
The way she was talking, it was as if she knew she should be afraid or worried about this. So did she remember her state from before whatever event led to this?
So one thing I should point out to you, some people see that and they think, oh, she doesn't seem very afraid. But remember, we're studying human beings here. We're giving mild shocks to the wrist where I ask you to set the level yourself so it's uncomfortable but not painful, right? So it's not like for most people they're like, oh my God, I was terrified. For her, we have done diary studies where we ask her on a daily basis to rate her emotional state. And she rates being anxious or afraid at the same frequency as individuals with normal brains of the same age. Right? So our best bet is just what she tells us. If she has to do a rating, she rates herself relatively normally. And that's, this is what you get with the subjective emotions, the feelings. So I can't tell from examining her if that's because she feels like she should feel afraid in that place, and so she rates the fact that she is, or if she feels the same thing that somebody else feels.
Yeah, but one is something that's learned versus her own experience.
Right.
And another is this, I guess, what I would call intrinsic emotionality about it.
Yep.
And those seem to me to be two different things.
But the only way we can actually know.
From what the clip you showed.
Yep.
Whether she remembers a state that was different than this.
We don't know, right? How could we know, right? If I ask her what her feelings are, and she tells me what she does what everybody would do, rate what her feelings are, she appears to be normal, right? But whether she's actually experiencing the same thing, who knows? All right. So this led us, I want to ask the question, does this simple model of fear learning apply to the types of fears that are typical of humans, learning through cognitive and social means? So you can imagine, translating back to our simple learning paradigms, here you could learn to fear this blue square because I paired it with a mild shock to the wrist. Here you can learn because I tell you when you see a blue square, you might get a shock to the wrist. And I can tell you the response I see, the autonomic response I see in this situation looks virtually identical. It would be hard for me to tell from looking at your body response which situation you were in. I could also say, watch this guy, because what happens to him is going to happen to you next. All right. So we've done both of those studies. And when I say to you, you see a blue square, you might see a shock. We see robust activation of the amygdala, specifically the left amygdala, and actually only damage to the left amygdala impairs the physiological expression in that case, consistent perhaps with the idea that this might be a linguistic representation, localized left hemisphere. In observation, I see robust amygdala activation both when you're watching the other person and when you think you might get a shock yourself. So this tells us, This tells us that these cognitive and social means of fear learning may rely on similar neural mechanisms. Now here, this is a lot of details here. I sort of put this up for the experts in the room, just to say that this is a model of fear learning from rodents. We've broken the amygdala down into different subnuclei. We know the lateral nucleus of the amygdala is the place where the association between the neutral stimulus and the aversive stimulus is formed. We also know that you can get this other representation, your knowledge, that depends on the hippocampus, that these two things are paired, that instructed fear, this can drive a response, at least the left hemisphere representation of that depends on the amygdala. And in observational fear learning, we see that regions of the brain involved in mentalizing about others and empathy actually correlate with the strength of your learning from other individuals. And we can sort of add these circuits to this simple model from other animals. And in this way, we can sort of build up from these simple models we can look at in rodents to the types of situations that are more typical of human experience. And here I just want to acknowledge Sumi Funiyama and Andreas Olsen, who worked on this when they were graduate students. Sumi is a professor at Gallaudet University, and Andreas is at the Karolinska Institute. All right. I want to now switch to the second part of my talk, which is not about how we get fears, and that was really probably my first 10 years or so of research looking at this, but what I've done for the last decade or so, which is once we have a fear, how do we get rid of it? And this, of course, is a much more clinically relevant question. People come into the clinics with a fear-related disorder, and our challenge then is really to help them figure out how to get rid of these maladaptive fear responses that they may have that is causing their disorder. And again, I'm going to focus on start sort of looking at things we can investigate in animal models and try to move to situations that might be more typical of humans. And here we're often talking about the relationship between the amygdala and the prefrontal cortex. And I'm going to just tell you what I'm going to talk about extinction learning, emotion regulation, using our cognitive strategies to reduce fear responses, the impact of stress on this, and then finally, a novel technique we've been looking at, reconsolidation. So just to start with extinction. So extinction, just again, as Pavlov described it, in this case, in relation to learning acquired fear responses, Extinction is the repeated exposure to a learned fear stimulus with no aversive consequences, resulting in a diminished fear response. We know from a number of studies that extinction learning results in a new learning that the stimulus is safe. So really what happens in extinction is you now have two memories. You have one, this thing is dangerous, and another one, this thing is safe. And what happens is they compete for expression. And we know this because in a number of circumstances, even after the fear is extinguished, the fear response can return. And extinction forms the basis for exposure therapy treatments of phobias and also exposure therapy treatments for PTSD. So that's illustrated here. You may not be able to read this, but it says, Professor Gallagher and his controversial technique of simultaneously confronting the fear of heights, snakes, and the dark. So I'm not a clinician. I think it's a little more subtle than that. But, so here is just again, starting with animal models, here is a sort of a diagram of the circuitry of extinction taken from research in non-humans, mostly rodents. And I'm not going to talk about this part of the circuitry, the hippocampus, but what we know is that the hippocampus is important in telling you that this is a context where that stimulus is safe, this is a situation where it's not. So for instance, you know, in a jungle, seeing a tiger is not safe, but at a zoo it is. So this is this contextual modulation of the expression of extinction depends on the hippocampus, and we have evidence that's true in humans as well. I'm going to focus on this part of the circuitry, which is here's the infralimbic cortex. In humans, I'm going to call this the ventral medial prefrontal cortex, because we can't really be that specific in humans. But what we know from research in rodents is that when you express an extinction memory, the infralimbic cortex inhibits the output of fear of the original memory, inhibits the amygdala response. So the original fear memory is stored in the amygdala. To express the extinction memory, you inhibit the expression of that memory to then show the evidence that extinction occurred. So we're going to look for evidence of this circuitry in humans. So what do? Well, we first have people acquire a simple fear response by pairing a blue square with a shock and a yellow square without a shock. We then, immediately after that, have a series of trials where the blue square is not paired with shock. We then bring them back a day later, another series of trials where the blue square is not paired with shock. This is my skin conductance response. The black line is the stimulus that was paired with shock, the conditioned stimulus plus, we call it. The gray line is the stimulus, this one that was not, the CS minus, we call it. So here we see robust acquisition of the fear response. Some evidence of extinction the first day. A lot of evidence that the fear memory returned, so we call it spontaneous recovery, and continued extinction the second day. Here I'm looking at the amygdala. So here's the blood oxygenation level dependent signal that I pulled out from the amygdala. This takes about 6 seconds to reach a peak. This is every 2 seconds here. Each scan was every 2 seconds. So here what I see in the amygdala during fear acquisition is an increase in bold signal in the amygdala, just like I showed you before. During extinction, a slight decrease in bold signal in the amygdala. And on the first day, this difference correlated with individual differences in the amount of extinction. When I looked in the prefrontal cortex, I'm going to focus on this region here. This is the ventral medial prefrontal cortex, the region that we think is most analogous to the infralimbic cortex in rodents. What we see in this region here is a decrease in the bold signal to a stimulus that's paired with shock that increases as extinction occurs. So this is early extinction, this is late extinction, and here I'm plotting these differences. So if I were to stick an electrode in this region in a rodent, what I would see is while extinction is occurring, I'd see an increase in firing rate. Here I'm seeing the same basic pattern, but it starts from a baseline of a decrease.
Do you have any idea which areas that encompasses?
Well, so we think this is analogous to the infralimbic cortex, but you know, area 25.
Several archetypal
excuse me. We think it's area 25 would be infralimbic. I mean, we can't really tell if it's infralimbic for sure. In rodents, the infralimbic and prelimbic are right next to each other. They're both area, but in humans, they think the prelimbic is moved up here. So that's, people generally think this is prelimbic in humans. And I should point out, the difference we see, the increase we see in bold signal in this region correlates with the retention of extinction on the second day. So this tells us, like animal models, perhaps not surprisingly, the amygdala is important in the acquisition, storage, and expression of this type of learning. And in order to express the extinction memory, the infralimbic cortex, the ventral medial prefrontal cortex, inhibits the amygdala response to express the extinction memory. So I want to move on now to another type of technique we use to control fear that's more typical of humans, and we call this emotion regulation. So I think one of the underappreciated facts about emotion is that we often think of our emotional reactions as sort of reactions to things that come at us. But the truth is, how we interpret a situation can have a lot to do with our emotional response. So, this is just sort of a glass half full. We can think of the glass as half full or half empty. This gets at the basic principle of some cognitive techniques for emotion regulation. So here, in this case, I'm calling it this technique for emotion regulation to reevaluate a stimulus associated with negative affect into neutral or positive emotional terms. And this forms the basis for cognitive therapy. So just to give you an example from my own life, if I were swimming and I happened to run into this, I could, being me, have sort of a exaggerated response about the danger. If I wanted then to try to control that in some way, I could go, what would my mother do? And of course, my mother loves all sea creatures, right? So if I could start to think about, all the things she told me, it can't hurt me, they're more afraid of me than I am of them, la la la, whatever she said, and try to reduce my emotional response. All right. So how do we do this using the simple paradigms that we're using in our laboratory to try to build up from the animal models? So again, I'm just going to stick with my blue squares paired with shock and my yellow square not paired with shock. And on half the trials prior to presenting the stimuli, we presented the word attend for a couple seconds. And participants were told, when you see the word attend, I want you to try to focus on your natural feelings when you see this stimulus. The other half of the trials, they saw the word regulate. And when they saw the word regulate, they were told to try to think of something, some image from nature that was calming to them, that the color of the square had reminded them of. And so we did this interspersed among in the paradigms, and we practiced this ahead of time, coming up with this image so they could do it rather easily when they saw the word regulate. We didn't tell them what the image should be. So if it was a blue square, it could be a water scene, a yellow square, field of daisies, whatever they felt was calming in nature to them. And participants could use this strategy. So here I've plotted the skin conductance response. Again, in yellow to the stimulus never paired with shock, in blue to the stimulus paired with shock. And you can see that they could use this strategy to reduce their arousal response to the stimulus paired with shock. So here I'm plotting what I saw in the amygdala. This is basically the same thing replotted on the right and the left. If we see here, there's a stimulus paired with shock. During attend, we see an increase in bold signal in the amygdala. During regulate, we see a decrease in bold signal in the amygdala. Just to plot this next to what we saw in extinction. So again, extinction, and when you're attending to the emotional significance, you see an increase in bold signal in the amygdala. When you're extinguishing or regulating your emotional response, you see a decrease. When we looked in the prefrontal cortex for regions that showed greater activity when you're regulating your emotional response versus attending, two emerged. One was the dorsolateral prefrontal cortex. We see the dorsolateral prefrontal cortex in a lot of studies of emotion regulation. We think this is important. This is a region often implicated in executive control, working memory. We think in this case it's important in sort of the online reinterpretation, the cognitive effort needed for the online reinterpretation of the meaning of that stimulus. And we also saw a region of the ventral medial prefrontal cortex. This is a region that overlapped with the region that we saw in extinction learning. Now we know that the dorsolateral prefrontal cortex does not have a lot of direct projections to the amygdala. It has stronger projections to more ventral and medial regions of the prefrontal cortex. So we thought that perhaps this region was having its influence on the amygdala through the ventral medial prefrontal cortex inhibitory circuitry that's important in extinction learning. So we looked at this region. We took this region from our extinction data set, and we plotted responses during extinction and emotion regulation. So here is when you have acquired during the acquisition of a conditioned response, and here is when you are extinguishing that response. Here is when you're attending to the emotional significance, and here is when you're regulating the emotional significance. And if we use this region and look for regions of the brain that correlate in response pattern when you're regulating your emotional response, two regions emerge, the dorsolateral prefrontal cortex and the amygdala. So this tells us what we think is that in emotion regulation, when you're using a cognitive strategy to diminish a fear response, the amygdala is important in the acquisition, storage, and expression of this type of learning. The ventral medial prefrontal cortex plays a role in inhibiting the expression of that fear memory in extinction, but also when you use a cognitive strategy, and here we use the dorsolateral prefrontal cortex to engage this same circuitry. And this is work done by Mauricio Delgado in my lab, who's now a professor at Rutgers. So what I've suggested to you so far is for extinction, to control a fear with extinction, the ventral medial prefrontal cortex inhibits the amygdala and the expression of the CS-US association, enabling expression of the new learning about the value of the conditioned stimulus, that it's now safe. In emotion regulation, this prefrontal cortex role in inhibiting the amygdala extends to the cognitive control of fear. And these are very effective strategies. They're used in the clinic all the time, but they're not perfectly effective. Like many people don't respond to exposure therapy or are able to use cognitive strategies. And some people can, and then something happens and all of a sudden they can't. And so part of the problem with this circuitry is that fears can return. And this was actually that was the premise behind this movie. I don't know if you saw it, but it says, just when you thought it was safe to go back in the water. We know through the passage of time, through changes in context, just even being re-exposed to the unconditioned stimulus again, the fears come back. Another time the fears will come back is when you're stressed. So if you've ever learned to sort of overcome a habit, you know that it's really hard to do that when you're stressed. And part of the reason this is hard is because we know that stress impairs the prefrontal cortex. And the prefrontal cortex, of course, is necessary to control a lot of these habitual responses, and also we think necessary to control fear responses. So that's what we want to investigate. So here is a slide from a review by Amy Arnstein. And stress has multiple effects on the brain and uneven effects on the brain. There's some brain circuits where it enhances, like the amygdala, sort of tends to enhance performance. But even relatively mild stress results in difficulty with prefrontal dependent tasks like working memory. And so we hypothesized that stress might impair the inhibition of fear. And again, even mild stress, when you do stress in a laboratory in humans, it's by definition mild. So we first asked, does stress impair the expression of extinction memory? We brought participants in the laboratory, again, had them acquire a fear response, had them extinguish a fear response on the first day. We then brought them back a second day. Prior to the second, the test of extinction recall and the second extinction session, we stressed them. And how we stressed them in this study is we stuck their hand in freezing water for 3 minutes. This is actually a quite reliable stressor. Sounds a little weird, I realize. But we measure cortisol before and after. You see robust increases in cortisol. Interestingly, one of the best ways to stress people in the laboratory, in case you didn't know, is to bring them in, give them a topic, and make them give a public presentation, and then look disapproving. Just saying. We don't do that because there's rumination and all sorts of other issues, but so we use this simple one. All right, so they were stressed or not. So the control participants stuck their hand in room temperature water. And then we looked for extinction recall on the second day and then did a second extinction session. So here I've just plotted, this is the stress response. Sorry, it's so small. The control group is in blue. The stress group is in red. This is subtracting out the baseline cortisol response. And here's prior to the extinction session. Here's after. We see a reliable increase in cortisol in the stress group, but not in the control group. Here, this is acquisition. I've plotted here the difference in the skin conductance response between the stimulus paired with shock and the one that was not. So anything above 0 is a conditioned arousal response. So here's acquisition. Here's the first day session of extinction. Here they come back the second day. They're stressed. And you can see we get greater extinction recall in the stress group, and they quickly extinguish again. We wanted to look now at a different type of fear control. So again, we're using an emotion regulation technique. Here, instead of blue and yellow squares, we presented pictures of snakes, one of which was paired with shock, one of which was our control stimulus. They were conditioned on the first day. Immediately after conditioning on the first day, they were given what we called a cognitive restructuring technique session. And this was actually, we worked with a clinical psychologist to try to come up with the type of technique you'd use in cognitive therapy that could be applied to our fear conditioning experiment. So here, after this, they were asked to look at these stimuli, tell us about the emotional intensity they had when they saw each of the stimuli during the fear conditioning session. They were then taught about the relationships between their thoughts and emotions, shown little cartoons like this, where here we see two cats looking at a dog. One cat sees it as a playmate, the other one's terrified. It's actually the exact same stimulus. So how is it your thoughts can actually contribute to your emotional responses? We then said, tomorrow you're coming back to the laboratory. You're doing the exact same thing. So what are you going to tell yourself tomorrow? How are you going to change your thoughts to change your emotions? They then came up with different strategies they could use. They could think, well, the probability of shock is quite low. In most of these studies, we shocked them about 1/3 of the time when they saw the stimulus. It's only a picture. The shock is really separate. The shock always ended with the picture, so they saw the picture first. They can think about, these pictures are kind of interesting. I know exactly what's going to happen tomorrow, so I don't have to anticipate anything. And then we brought them back the next day, stressed them or not, measured cortisol, and then did the exact same procedure. So here I'm plotting on the left. This is the increase in cortisol from baseline. In the stress group, we see robust increases in cortisol. No increase in cortisol, the no stress group. Here, this is the efficacy, so how much were they able to reduce their conditioned response on the second day. Here I can see that the participants in the no stress group could use this cognitive strategy to reduce their emotional response, whereas participants in the stress group could not use this strategy to reduce their conditioned arousal response. And these participants, they felt like they were using it in the same way. These tasks occur at least 10 to 15 minutes after the stressor. So they don't actually feel that different at that point. Nevertheless, they're unable to use this strategy to diminish their arousal response. So we think in stress impairs extinction recall and emotion regulation, perhaps by altering prefrontal cortex function and making this inhibitory circuitry less capable under stress. We haven't done the imaging studies on that yet. We're working on that now. And this is Candice Rao. Candice is a graduate student in my lab right now. So what I've suggested to you so far is that we have a lot of techniques for diminishing fear, but a number of circumstances, including relatively mild stress. So again, this is not a robust stressor. It's not a persistent stressor. It's a relatively mild stress. can impair your ability to use those techniques. So what we'd really like to know, is there now a way that we can diminish fear without having to use the prefrontal cortex to control the fear? In other words, is there a way to persistently block fear so the fear does not return? And to look at this, we've started to examine this process called reconsolidation. So I'm going to walk you through a little bit what reconsolidation is and then tell you about some of our studies. So reconsolidation is this idea that a previously created long-term memory is brought back to a labile or fragile state by re-exposure or reactivation of the memory. So just to give you a little more background, so the typical view of memory, over the last 50 years or so has been this view, which is you learn something, there's a period of time that's required for all the synaptic changes that become that memory, for that, and that's called consolidation. So you learn something, there's this consolidation process, it takes some time. If you were to do something right here after you learned something that disrupted the consolidation process, you wouldn't store the memory. So if you know somebody who has a concussion and they don't remember anything that happened, it's been argued that that's because it disrupted the consolidation of those memories. But it's been previously thought that once you store a memory, that memory is somewhat stable and fixed in your brain. You can have forgetting over time, you can have interference, but the basic characteristics of that memory stays the same. And if you retrieve that memory, you retrieve it, this sort of same memory trace. The next time you retrieve it, you retrieve that same memory trace, and so on. The reconsolidation view has been, over the last 10 years, there's been a lot of interest in this idea that it's a different way that memory works. So you learn something, you store the memory, that it takes some time for the synaptic changes that become the memory to occur. When you retrieve that memory now, there has to be another storage process. Not identical to the first storage process, but a second storage process that also requires synaptic plasticity that takes time. So then every time you retrieve the memory, you're actually then restoring the memory. And if this is the case, then this tells us there's a second opportunity now where I could do something to change that memory. I can prevent not the storage of that memory by disrupting consolidation, but I can prevent the re-storage of that memory by disrupting reconsolidation. And this, in the clinic, you think about this gives us the idea that maybe something, that happened a long time ago, I can maybe selectively disrupt that memory at a much later time. So this is the idea behind reconsolidation, and this notion really took off in the year 2000. There was a paper published by Kareem Nader, who was working in Joe Ledoux's lab at the time, where he did something like this. He brought rats into the laboratory. He played a tone. He paired it with shock. That was the learning phase. He waited for that fear memory to consolidate. He brought the rats back, played the tone again. Then he injected into the lateral amygdala, where we know these simple associative memories are stored, a protein synthesis inhibitor that disrupts the synaptic changes that become that memory. And then he found later, when he played the tone, it was like the rat had no memory of the fear itself. So we were very excited about this work. We thought, well, can we find evidence for this in humans, this mechanism of reconsolidation? The problem is, of course, you can't do this in humans, right? You can't inject protein synthesis inhibitors into the brain. Don't try it at home. And so we had to think about whether a different way we could get at this. So if you think about reconsolidation, if this process is real, if this is how memory really works, reconsolidation, this notion that we restore memories when we retrieve them, didn't evolve so that we could inject drugs into the amygdala and disrupt memories, right? It evolved for another reason. One reason could be this notion of updating memory. And this is actually an idea that was very big in cognitive science in the 80s with eyewitness investigations. When you question somebody about something that happened and you give them new information, does that actually become part of the memory itself? But there was no neurobiological model for that, so there was really no resolution to that debate. But if this is one of the purposes of reconsolidation, the other purpose that's been argued is to strengthen memories. So if you test and retest over time, every storage process strengthens the memory. But if this is the case, and we can update the memory with new information about the value of the stimulus, maybe we can take advantage of this function to permanently alter a memory, of a fear memory. And so the way we did this, I'm going to show you one study that we did. We've done a number at this point. We brought participants in the laboratory. Here we presented two squares, both paired with shock. So here a yellow square paired with shock, a red pair paired with shock, and a blue square, again, our control stimulus. We waited a day for that memory to become stored, consolidated. We brought them back a day later, showed them just one of these squares paired with shock. We waited 10 minutes. Now this 10-minute, this is going to seem odd to you, but this 10-minute break is critical sort of for the reconsolidation process to start. And we can talk about what we think some of the reasons are in the discussion if you want. We waited 10 minutes, and then we simply did simple extinction training. Now, extinction training, remember, is safety training. I'm now training you this stimulus is now safe. The only difference, and we presented all three stimuli here, the only difference is for this stimulus, which I call CS plus R for reminded, we're doing extinction training during the reconsolidation process while that memory is being restored. For this stimulus I call the non-reminded, we're doing standard extinction like I just showed you. Okay, so that's really the difference between the two. So the hypothesis is we're updating the memory in this case because it's this information is coming during the memory restorage. And here we're creating a new memory. Yes?
Sorry, just to be clear, so there was no shock during the CS plus R reminder?
No, there was not. And just so you know, there was one less presentation here to keep the number of presentations equivalent. All right. We brought them back a day later. We gave them 4 unsignaled shocks. This just is to reinstate the fear memory. And then we do a second extinction session here, looking at the beginning of this process to see did the fear return. So here's our data. So just so you know, we only selected people that showed equivalent fear acquisition to the two stimuli. That's why this looks so pretty. Because if they don't acquire similarly, we can't look for differences later on. So here is the the skin conductor response to the stimulus, two stimuli paired with shock versus the baseline stimulus. Here's the end of extinction. So they extinguished responses to all three stimuli on the second day. Here's the third day. We do 4 unsignaled shocks. And here's the early trial of the recovery test. So the fear comes back. As we would expect with an extinction memory, the fear does not return when we did extinction or safety training during the reconsolidation process. And here we just extinguished before they left. In another version of this study, we actually brought people back a year later. And we found that the fear would still come back in the participants who got standard extinction and did not return in the participants who got extinction during reconsolidation. It was simple extinction training. We did the exact same thing we did previously, right? But the only difference is that we did it either, we believe, during the reconsolidation process or not. And this 10-minute break here is critical for that. And research in rodents has shown that, if you wait, you have to wait at least 3 minutes to begin that process. Do you want to?
Something about how you know maybe about
Let me do this at the end because it's kind of complicated. That's okay? All right. So just looking at the brain, what are regions? So now, just so you know, for those of you who, I'm looking at brain responses during extinction. Okay, so what's different during extinction? The only brain region that emerges as different between the two stimuli paired with shock during acquisition is this ventral medial prefrontal cortex region. And this is, here I'm plotting the results during the early part of extinction. So if you remember, we get this decrease in the bold signal during early extinction that increases during extinction training. So here's early extinction. I see greater responses to the stimulus that got safety training during reconsolidation than the one that did not. And here I've plotted that bold signal. So here is that decrease in bold signal that I showed you previously during early extinction. We don't see that decrease during extinction training for the stimulus that's getting safety training during reconsolidation. Here I'm just plotting that difference. Remember I told you that it goes up during extinction training. Here's early to late extinction differences. So here we see no change over extinction for the stimulus that underwent safety training during reconsolidation. And here for the standard extinction stimulus, we see that increase again. And again, nothing for our baseline stimulus. And if we look at connectivity between the amygdala and the ventral medial prefrontal cortex, we see much stronger connectivity for the stimulus undergoing standard extinction than the stimulus that underwent extinction during reconsolidation. So this suggests to us that extinction during this restorage process may have led to re-encoding of the memory as safe, that the behavioral interference of reconsolidation diminishes involvement of the prefrontal cortex. And then what we think is that but we haven't proven this, but we think that the fear memory no longer exists in its original fearful form. We've kind of rewritten that memory. And I should point out, this is work by Daniela Schiller, who is now a postdoc. Excuse me, she was my postdoc. She's now a professor at Mount Sinai School of Medicine. And Jonathan Cohen, who's now a medical student. I'm sorry, Jonathan Kanan. Sounds like Cohen. So I always, when I present this work, I always, want to give a little caveat because, because when I talk about this, in the press, people often think, oh, you have a way to treat fears. We've only done this in simple associative fear memory at this point. As I've already emphasized to you, human fears are much more complex. We have many different forms of representation. We really have no idea at this point if this sort of reconsolidation interference mechanism is going to be useful in treating the complex situations and fears that are typical of human anxiety disorders. The only published clinical evidence that this type of thing might be a more effective treatment has come from research in addiction. So there was a group in Chinese that looked at drug addicts and showed that if you essentially did a reminder prior to standard exposure therapy for cravings, you had more persistent reduction of cravings. So that's the only clinical evidence that something like this might actually be useful so far. So just to sum up this final part of the talk, what I suggested to you is that during extinction, the ventral medial prefrontal cortex inhibits the amygdala and the expression of this fear memory to allow the expression of the extinction memory. During emotion regulation, the prefrontal cortex role in inhibiting the amygdala extends to the cognitive control of fear. That stress and other things impair this fear inhibition. And for stress, it's perhaps by altering prefrontal cortex function, and even relatively mild stress can do this. But by targeting the reconsolidation process, we think what we're doing is changing the original fear association, and this then reduces the involvement of the prefrontal cortex in fear inhibition to reduce the expression of fear. So what I hope I've proven to you today, or at least demonstrated to you today, I should say, is that these animal models of fear conditioning can provide a basis for understanding human fear learning. But I think it's important to remember that we have many other ways to acquire fears. We have a complex social environment and symbolic means of communication that give us greater flexibility in our fear learning system than other species. And that this flexibility of human fear learning can provide unique challenges when we try to translate this research from animal models to the treatment of clinical disorders. So I want to thank the people that paid for this work, the National Institute of Mental Health, the James S. McDonald Foundation. I showed you many of the people who did the work, but other people helped with the work and inspired the work. And I want to thank you for your attention. Thank you. So wait, we had a question about the 10-minute window, right? Should we get to that? So we don't know, right? The answer is we don't really know. There have been some work in rodents where they did the reminder and then the first section 3 minutes later versus I forget, and that study was an hour, and showed changes in the lateral amygdala indicative of synaptic plasticity only when you did it later, when you waited more than 3 minutes. There's some evidence to suggest people are starting to use learning models looking at things like prediction error, so maybe it's really something related to the predictability or something. I don't really know. This is, I think, a hot topic of investigation, which is essentially what is it that takes a memory from one, this situation from now I create a new memory to now I'm updating the old memory, right? Because that's really the switch we're talking about. All we know is this 10-minute window works. In rodents, they usually wait an hour. We have a control where we wait 6 hours. That doesn't work, right? So this window, we call it, closes somewhere between an hour and 6 hours. But we don't actually know, is it really time per se, or is it something about the environment that tells you that this is a situation where you should update that memory and this is one where you should create a new memory? We don't know.
So, and this is what you take to be why you're in the reconsolidation period, and the part of the puzzle is that when you're actually doing the extinction.
The first trial is just like a reminder, absolutely.
So when you're doing the normal extinction case,
yep.
Are you not bleeding over to the 10 minutes? It's basically all over by the time that memory is.
Well, the notion is once you start creating that new memory, you're just adding to that new memory, right? So it's not, so now you're not, it's not like, it's one or the other. You're either sort of creating a new memory and you're adding to that one, or you're actually.
Start the extinction, and now you're still working on the same one.
Yeah, so I mean, so that's the notion, right? But all of this is really, this is brand new research, right? So we're really trying to figure out what is it? What creates that transition? And what we know is this works, right? But we don't know what is the principle, is it a time per se, or is it something about the environment, something like prediction error of some sort of sociability that's now creating that transition from sort of an old memory to a new memory, or from, updating an old memory to creating a new memory. Dan, you had a question? And then I'm going to take questions for students before I go back to speakers.
Fascinating talk. Thank you very much. Makes me wonder if anybody studied amygdala responses in two different groups of human subjects. Those who love to go to scary movies and those who some people seem to be sort of addicted to hitting their amygdalas. And I wondered if over time they develop what you might call an abnormal fear response from their addiction to the movie.
Every Halloween I get that question from reporters and I never have a good answer. It's hard to say. I mean, I don't know. And I should say that, we talk about the amygdala as involved in this fear response, and it is, but we see amygdala also when you get, highly arousing positive responses as well. We know that, that there's some different circuitry involved from the work in rodents with appetitive and aversive conditioning, but nevertheless, it's not just, it's not just in this case that we see amygdala. But, I don't have an answer to that question. I don't know anybody who's done that study. And I disappoint reporters every Halloween. In fact, I get, people ask me to be on radio shows, and before I say, look, I'll come on your radio show, but if you ask me that question, I'm going to make you disappointed, because I don't have an answer. So, yes.
Right. When you were talking about the reconsolidation, I was wondering if you thought about the consequences of fear. So in your example, when I think about a shark, okay,
yep.
Shark is scary, yes or no?
Yep.
But suppose I have had this experience. Suppose I go, the shark eats my leg, or eats my mother, and then I end up in an orphanage, and
yep.
I don't know, something about this
yeah.
happens. So there is a consequence to that fear.
Yep.
So when you interrupt that, so let's say you reset the amygdala in that sense, but that's the fear of shark, but there was a lot of
yep.
consequences.
These are the questions that, so first of all, I should also say, these participants also had perfect knowledge of what happened, right? So we were really targeting, remember in the beginning I showed you the amygdala isn't necessary for telling me what happened, right? So it's not that there isn't some still storage of that going on. And, and something like a real life human fear, that's what I was mentioning at the end, right? Yeah, okay. We don't know. We don't know if we're just, if all these, if you have multiple representations. In the one study they did with addicts, where they did this, where it's targeting an impetitive memory, they still did many trials. They just found it was more persistent long term. The fear didn't come back or the craving didn't come back so much, right? So this is going to be one of the challenges: is how do we get at these really complex situations that may have multiple representations? How do we know we're targeting that specific memory, not another one? If you do the task, so some of the studies in rodents, where they're injecting drugs, they can also disrupt an extinction memory. So then you've created a whole other situation.
On the other hand, do all fears have some conditional stimulus attached to them? Should they have necessarily stimulus attached to them?
Well, so as I mentioned, the germs, right? You don't actually, you know they exist, but they only exist in your head, right? I mean, they exist. We know they exist, right? but you have no perceptual experience of that. So as far as you're concerned, it's an imaginary fear, right? But supposedly it's a real fear. So, yeah.
You may have sort of just briefly sort of touched on it in answering the last question, but did your participants in the reconsolidation experiment behave like the amygdala patients? You know, in that, did they have that dissociation subjectively of saying, I know something?
So, you know, so she looks like, I mean, yes, they did know that something, they did know that was happening. But if you imagine it, what's weird is, right, is that the fear actually comes back with regular extinction. Because even when it comes back, at least in terms of arousal, they already know that they're not getting the shock, right? And so it's almost weirder that in the extinction folks it comes back, right? Than the sort of that it doesn't come in that regard, because they all have the same knowledge that things had changed. And so, you know, and we think that maybe is, you know, some notion that these two systems, one sort of cognitive and one, you know, one more implicit, you know, actually, you know, are not completely in sync always, right? Yes. So this particular patient, we've done this with many patients. Most of them have unilateral amygdala damage, and that is from the result of surgery for epilepsy. She also had epilepsy. She had unilateral temporal lobectomy on the right, and she also had scar tissue in her amygdala throughout the left amygdala. So before they did the temporal lobectomy, they did a biopsy because they were a little concerned. I actually met her. The neurosurgeon called me and said, "What happens if I take out, you know, this side when I know that the other side is impaired?" And at the time, we knew almost nothing, right? She chose to have the surgery anyways because, you know, epilepsy was ruining her life. But she has scar tissue throughout the left amygdala and a unilateral temporal lobectomy on the right. So her damage goes beyond the amygdala on the right.
But it was all, like, fear response, it was all lost at the same time, like abruptly, rather than...
No, so we've actually done some studies looking at patients with epilepsy prior to the surgery. And I should point out, unilateral temporal lobectomy, unilateral amygdala damage is enough to show an impairment in fear conditioning, and most of the things that we show impairments are with amygdala damage. So what's interesting, the hippocampus is kind of like half a hippocampus is almost as good as the whole hippocampus. With the amygdala, it's like half an amygdala is almost as bad as no amygdala. And so with her, and these patients, when we tested them prior to surgery, at least on fear conditioning, they don't show normal condition fear responses prior to surgery. Now, often, if this is the foci of the epilepsy, that tissue is not working normally prior to having it removed. So we think the damage is actually already there. And then the question becomes, and I think this got back to some of the questions you had about her feeling, you know, so some have argued that individuals who develop amygdala damage later in life have different patterns of emotional responses than individuals who have it earlier. There's some really nice work by David Amaral on monkeys showing that's the case. So the consequences are different. You know, having a lifetime of experience that with an intact amygdala and then not could be different than having it before. Yes?
So you mentioned the idea of a prediction error related to the reconstruction.
Perhaps. You know, we don't know, yes.
So in that sense, is it relevant that the first trial on the reminder day was not paired with the US? Because that would be...
So some people have, so there's been a lot more research in rodents than humans. And some people have shown that you can actually just do a US to remind to do the reminder, and it's actually more effective if you do the US. So, so I don't know if it's relevant that it was or was not.
Yeah, okay, but there wasn't a version of the study that had a CS US.
Not in ours. That hasn't been done in this paradigm in humans, but in rodents, yes. Yeah.
So what I'd like to follow up with that, you'd shown earlier that it can be attributed to dorsolateral prefrontal cortex activation being able to mediate ventral medial effects.
We think, yes. Mm-hmm. Well, actually, no. I think it's only in regulation. I don't think you need the dorsolateral prefrontal cortex for extinction.
But to regulate the effect
in cognitive regulation. Yeah. Well, I think it's for when you cognitive regulation is what you're engaging that circuitry. Yeah.
And so, in working with humans, that's kind of how you ended it with how do we build up to more complex.
Yep.
What happens if you don't even plug in for the SCR for the shock?
Mm-hmm.
So the equipment isn't even there, and you just have the CS.
Yeah, I don't know.
Full
We haven't.
Well obvious to them. There can be no shock during that reminder
Yeah,
period.
we haven't tried it.
I'm curious.
Yeah, haven't tried it. Yes.
So a question about the extinction regulation about the amygdala change in
Yep.
translation. One of the questions that just came up a second ago was whether the dorsolateral prefrontal cortex is working through
Yep.
the ventromedial. And presumably the effect of the activation changing the amygdala is presumably maybe coming from the central amygdala, right? Because the model, at least that comes from the rat, is that the inhibition is taking place in the central amygdala. Not
Well, yeah, the inhibition is happening between the lateral and central. Yeah.
Interrelated.
Yeah.
But so have you tried to look at whether there's any sort of spatial distribution voxel-wise?
We haven't. We haven't. And part of the reason is, I mean, we'd have to do a high resolution study to do that because, in humans there's a 1 millimeter difference between the central and nucleus. I mean, the central and lateral nucleus. And so, and if you do, so the amygdala is quite small. For those of you who don't know, it's about the size of your thumb tip. And so we don't actually, when anybody tells you they can actually differentiate lateral from central in imaging, I don't believe them. So we have to do high resolution. We tried to do just that study.
The comparison between the regulation and extinction?
No, we haven't done that. We tried to just do extinction to see if we could separate lateral and central with high resolution. The problem was, and I think we've actually started to model the trial by trial amygdala response to get at this, is that it's a learning signal, right? So for bold, you need to group across a number of things to see something. And if it's a learning signal, by definition, it should be changing over time, right? And so, we do partial reinforcement in part to slow down the learning so that we can actually get a bold signal. But I think we've recently come up with a model of associability that seems to track amygdala trial by trial. So, we want to try the high res again, using that to see if we can actually model the learning in the amygdala.
Yeah, I respect it's methodologically difficult, but I do wonder if without trying to put the delineations between central and lateral, artificially, if you just saw any distribution difference.
We didn't really, but I think what we're getting in our bold signal is sort of a compilation of both. And when we reduce one, we don't see much of anything, right? Because when we do extinction, we don't see anything. Theoretically, we should still see lateral, right? But we don't, right? So I think what we're getting in our bold is just sort of has to kind of be a big response to see anything. Yes.
Extinction or the
Yeah, the interference effect, whatever you want to call it.
Is it a single mechanism or is it tied to the memory? So in fact, what happens if you wait your 10 minutes and then start doing the procedure and interleave in that a recall for another memory and try to extinguish that without the 10-minute gap?
So that's kind of what we did in the sense that we conditioned two things simultaneously and we only reminded one, right? And then we extinguished both simultaneously and we only saw the difference in one. So that was a little specific.
Not quite the same thing.
Okay. So give me an example of the experiment you're talking about.
Condition 1.
Yeah.
Condition 2. Separate events.
So separate events. Okay. Yeah. And then
Walk a recall
on one of them and see if you get.
Wait 10 minutes. Start extinguishing. Provoke a recall on the second one.
And see what happens.
Switch to extinguishing it immediately.
Yeah.
And see whether it's a mechanism.
I don't know anybody who's done that or anything like that. Yeah, I don't know. I mean, I have to say these are very challenging experiments to do in humans, right? You have to bring them in 3 consecutive days. So, we've done studies where we've waited a period of time to see if we can still disrupt the memory at all, and things like that. But we've done. It takes a while.
Straightforward, not easy.
Yeah, not easy. Yeah. Okay.
Straightforward means 4 pages of
Yeah.
algebra.
Okay.
So do you want to take? So we can maybe we can take a break as a large group, and Dr. Phelps is here through lunch period, and so some of you can maybe find her on the break, but we can all stretch our legs, and then we'll reconvene in the big room here at 10:45 to begin with our next speaker. So thank you very much, Dr. Phelps.